Hi, my name is Etienne from the Dietrich Lab at Heidelberg University Hospital. I'm going to introduce you today to the next generation iRNA database. With Sci-ModoM, we're trying to address some of the pressing challenges that we face now in the epitranscriptomic community. I've already had a really nice pitch, so I think that some of these challenges were raised at different locations, and particularly recently in the United States by the national academies. And for those of you unfamiliar, I'm going to give you a bit of a background. I'm going to tell you what Sci-ModoM is and what part it can play in a larger RNA modification program, and give you an outlook on our current development roadmap. So our motivation here is the fact that when you consider the landscape of databases, those dealing with next-gen RNA modification data, well, the state of the art doesn't really reflect the state of the field. We have a mix of high and low resolution data. There's a lot of tailor-made processing. We have issues with lineage and traceability, much bigger issues with open standards, open science standards in general. We have data is just not findable, right? It's difficult to reuse, and there's no standard for data sharing. So obviously we want to improve data verification. We want data to be accessible as soon as possible. We need to engage with the community. This must include industry. That's really important. And if we really want to make an impact, traversing field boundaries, right, open new avenues in RNA research, we need better detection methods. We need more robust detection methods. And for this, why it needs to be started with open science standards, and we need to agree on common standards. So in particular, to address these challenges, we need single resolution, quantitative, per site and per data set. We need this kind of evolvability, right? We need continuous updates. We need to obviously adopt open science. We need to eliminate this kind of tailor-made processing. We need more user control. And it doesn't mean only more a best user experience. It means things like simple as stop pre-curating evidence, leave the user all the data, but that also means that we need infrastructure for that. So we think, all in all, that we need a next-generation database, and we think that Sci-ModoM is partly addressing these challenges. At least we hope that we're in the right direction. We do that by incorporating reliability scores and stoichiometric information per site and data set, allowing users to keep full control. This can be done by looking at a set of combined filters, such as coverage or frequency. One can look at how many data sets actually support a given site across different organisms or different technologies. The metadata is easily accessible. In a click, you can actually download a given data set in a standardized format, which I will describe in a minute. This is fully searchable, and data sets are grouped into studies, and which are assigned permanent IDs. And this overall study metadata is also easily accessible with links to open-air repositories. And there's additionally an additional sort of layer of information where read-level information can be linked to the site-level information that is included in the database. The data can be continuously added to the application. Users can suggest projects, data sets can be added, and this is possible thanks to standardized format, the use of ontologies and nomenclature. And I would say here that the most important thing here is that there's no tailor-made processing in Sci-ModoM, so users can relate directly the primary data to what they find in the database. This brings me to the better armored format. So that's a format basically that extends the ENCODE format for RNA modification. It's still evolving. The latest version has been developed in collaboration with Oxford Nanopore, so it should be part of the official specs in some coming release of ModKit. We're sort of working with them at the moment. And in essence, that's a metadata header that includes some standardized fields with controlled vocabulary and additional free text fields with some recommendations that are not shown here. For example, the basic bioinformatics workflow could be the ModKit call used to reproduce the data, external sources, recognize IDs, experimental link to an open-air, and things like that. So there's still work to do. We're trying to improve that so that it becomes even fairer. And the data section basically is just a standard bed file, but we've sort of formalized the type and the range of the fields. And in Sci-ModoM, for example, score is fixed. So it must be what we call the valid coverage. And this is something that will be output, for example, by ModKit in the next release. So I think all this really is just the foundation, right? It's just the premises for a much larger RNA modification program. We need to sort of work collaboratively. We need to get involved in different aspects. And in particular, in our case, we're really sort of pushing this RNome project where the aim here is to sequence all human RNAs and their modifications. And we're working as well in collaboration with MoDomics, where we want to take all this information that is available in Sci-ModoM and move it to distill it to a more sort of sequence level information through MoDomics and eventually possibly in RNA Central for the use of the community. Obviously, it would be unfair to say that we're already there. There's still a lot of work to do. We're sort of integrating the latest specs into the database, adding an API documentation. We want to add more organisms, more RNA types. And also, I mean, recently I had a discussion with Blake. We just had a really interesting talk. He made some suggestions, which we're sort of working on. I mean, he's an expert in database. We're sort of not so much expert. We've been starting to work on that only a few years back. So there's still a lot of work to do. And I think, like, I ask you, if you're interested, really check it yourself. And if you have any suggestions, criticisms, if there's data you want to see in there, please come to see me or contact me afterwards. And we really hope to sort of work collaboratively with the community to make this tool useful for everyone. And with that, I would like to conclude. Thank RMAP for the funding, my group, the RMAP Scientific Board, Janos and Andrea, and Art and Marcus at ONT, and another bunch of people at RFAM, the Human RNome Project, and some other people who have shown some interest and supported Sci-ModoM, and you for your attention.
Questions for Etienne?
Hi. I was wondering if there is any curation going on where you get data sets from the literature or, available sources to integrate, or do you rely on outside users to submit their data?
So at the moment, the idea is that Sci-ModoM really is a resource of high throughput data. And the idea is that we don't want to pre-curate the evidence. So obviously that means that there's going to be a lot of data there. We're going to need to optimize the queries, but the idea is that the user wants to find out whether there's any modification at a given place in a certain RNA. Then it sees everything that's available, and he can eventually prioritize himself by looking at the score, the coverage, or the frequency, looking at how many data sets provide this evidence, and he can then sort of does the curation himself on the fly, right? What we are actually doing now is we're working in collaboration with MoDomics, where we want to take all this evidence in Sci-ModoM and sort of distill it in a way that MoDomics has some sort of a criteria, right? That they look at the evidence based on how many data sets produce this evidence, and they have like this set of criteria that they use. And we want to actually take all this evidence there and summarize it at the sequence level, so that in MoDomics it could be available at the RNA level. You would see the modifications, but only those that passes those filtering criteria. But that in Sci-ModoM, we really want to leave everything available for the user.
Maybe I used the wrong word there with curation. It was more a question about, do you actually look for the data and get outside data into the database yourself, or do you solely rely on submissions for that?
Okay, if I understand correctly, so basically the data that's there at the moment, we mostly put it there based on what was available in the literature, but we're sort of hoping that eventually we continue doing that, but that user actually may be able to suggest themselves, right, a project. So the idea is that you can register if you're interested, and then you can basically create this project, and then once it's accepted, you can upload the data yourself. So eventually it sort of be self-growing in a way.
Maybe I can ask a question. Does the database have information about the proportion of molecules that are modified? Like in ribosomal RNA, the sites are not, they can be fully modified, so all molecules have it, but they can be like 50% modified?
Yes, so this bit-RMOD format basically has this column, which is the frequency, which is basically the stoichiometry. So and that's why we're actually really pushing this forward, because there's been so many data sets that have been published that we cannot put into Sci-ModoM because they're not quantitative. And so I think that the field has to change this mindset, right? We need to produce quantitative data, and this bit-armored format really is where people should produce data where they have stoichiometric information per site, so that then this is available for the user, and then people can say, right, you know, this is a site, but maybe there's not enough coverage, or maybe the stoichiometry is not enough, and then, but yes, that should be there.
Okay, thank you very much. We'll move on to the next talk.